couple of reminders. Okay, your microscopy lab that we did on Monday. Okay, that is going to be due on Tuesday of next week. So make sure you're working on that. Okay, over the weekend. If you want me to review it, okay, bring it to me on Monday, either before school starts, at lunch, at nighttime, okay, or after school, and I can have a quick look at it and give you some feedback. It is due at 8:30 a.m. on Tuesday morning. And your toxin project, same deal, just Wednesday, 8:30 a.m. Okay, make sure you've got both of those things done. You're at school on Monday, but you could bring it to me tomorrow, and I could look at it then. Okay, if you wanted me to review it, but we don't have school on Monday. Okay, and so I won't be here to look at it because we have PD. We have to go somewhere. You guys get the day off. This is like a crap deal to me. Anyway, sorry. All right, so that's why I keep thinking there's actually school money, because I have to work and you don't. That is a crap deal. It doesn't just seem like a crap deal. Okay, all right, so we're going to look today at the parts of the cell and what they do. We labeled our diagrams yesterday. It might not be a bad idea to have those out just to kind of refer back to, but I do have pictures of all the individual organelles that we'll be working from. But this is where we learn what each part does, because we got to know that. All right, you're going to get questions on quizzes and on exams that are, here's a diagram of a cell. Tell me what this part is and what it does. All right, you're going to have to be able to do that. And then near the kind of second or last third of class, we'll say, we're going to start talking about how cells transport materials, okay, within themselves and across their membranes. All right, those are important things that cells must do because they're one of those basic functions that cells have to carry out. All right, so first key point, we need to understand the significance of compartmentalization in cells. All right, that's going to be an important term. Okay, if something is compartmentalized, it means that the different parts of it are separated from each other. Okay, our school is compartmentalized in a few different ways. Okay, most of the math classrooms are down the same wing. Okay, but each teacher has their own classroom within that wing. Okay, their little compartments is compartmentalized. Okay, the computer labs, they're kind of all together. Okay, and they're in their own place and there's separations between them. Okay, and what that allows us to do is to make sure that the stuff that's needed for a certain subject area is in one place where it can be the most efficient. Okay, it would hardly make any sense for me to have one desk in this classroom labeled math, one desk in this classroom labeled English, and one labeled social studies, and another one that has a computer on it, and another one that has a bandsaw, okay, for construction, right, and another one that's got an easel for art. Okay, how am I going to do all of that? Okay, that's not compartmentalized, that's generalized. And I have no idea how to teach English or art or social, okay, or even math. Okay, like it's not, I'm not going to do a good job of it. Okay, we want to be compartmentalized. Everything gets taught in this room has to do with science, and I know that stuff, so I can do a good job of it. Okay, whereas if it was everything all in the same room, it wouldn't work. Okay, or maybe a different example might be in your house, you have 3 different temperature environments. Okay, you have your living area, which is probably somewhere around 21, 22 degrees Celsius. You have your refrigerator, which serves the purpose of short-term storage of food, which is probably about 4 degrees Celsius. And you have your freezer, long-term storage of food, probably about minus 15. Okay, those are 3 very different jobs that are not compatible with each other. Okay, if I try and do all 3 of those jobs in the same place, what temperature do I have to keep my house at? Like 8? Okay, I keep my whole house at 8 degrees to find a happy medium between all 3 jobs. How good a job do I do of any of them? My house is 8 degrees, I'm cold. Okay, my food's going to spoil because it's too warm for it. I can't freeze anything at 8 degrees. Okay, so I'm going to have a house full of rotten food, and I'm going to be cold and miserable. Doesn't seem like a very good way to do things. Okay, so compartmentalization is an important feature of eukaryotic cells. That's the kind of cells we have. Every organelle that we listed yesterday on our cell is a separate compartment, because each one of those organelles does a different job that's not compatible with the conditions another organelle might need to do its job. Does that sort of make sense? Okay, so that makes the cell much more efficient, which is why eukaryotic cells tend to be a lot bigger, right, than prokaryotic cells, which are like bacteria, which are really, really small and don't have compartments. Okay, so they're not as efficient. All right, knowing the role of each organelle. That's just something you're going to have to memorize, basically. So if you're a person who likes having like, the recipe card kind of things, you can draw the picture and put the function on there. That's a great idea for this stuff, okay? Because this is memorize and regurgitate, okay? That's this part of biology. Learn it, know it, write it down, okay? Understand that cells are a complex system of interdependent processes. All right, so lots of things are going on in the cell at the same time, okay? Interdependent. Does that mean they work separately or they work together? Independent means work separately. Interdependent means they depend on each other. Okay, so while those processes might happen in different areas, they are needed for each other. So one of them might produce the raw materials for another one. And if that one breaks down, the next one in the chain breaks down, the next one in the chain breaks down, and you got a big problem. Okay, does that sort of make sense what interdependent means? All right, understand how the cell carries out its functions. So we'll be looking at, again, what do each of the parts do, how do those things work together, and we'll be looking at, again, transport across the cell membrane and within the cell. So unfortunately, today is going to be a lot of me yakking and a lot of you listening. Okay, sorry about that. Some days are like that in biology. Many days are like that in biology. Okay, so compartments. We saw this diagram yesterday, comparison of the plant and animal cell. They have a lot of similar compartments. Okay, the biggest compartment, okay, within the cell is probably the nucleus. Okay, and in there, that's where we keep all of the DNA. Okay, we don't want anything else to get in there because DNA is fragile. It can be easily damaged. And if your DNA gets damaged, there might be mistakes in it. And if there's mistakes in it, then you won't make things correctly and the cell could die. Okay? So it's got its own separate compartment where the conditions for storing DNA are ideal. All right? Your mitochondria, okay? That's the powerhouse of the cell. That's where cellular respiration, the energy yielding reactions go on. Okay? It needs very different conditions than inside the nucleus. Okay? In fact, that organelle tends to get kind of hot. Okay? And it's got probably kind of hostile conditions within it because essentially there's a combustion reaction going on in there, okay, all the time. You definitely wouldn't want to put things that are fragile in the same place that's going on, right? So that's what we mean by compartmentalization. Each one of these things has its own unique environment that makes it possible for them to do the job best. Okay, so cells are divided into compartments by membranes. Each one of those organelles has its own membrane on the outside. Some of them even have double membranes, okay? So that it's present only in, as we said, eukaryotic cells. Okay, eukaryotic cells have the compartments; they have a nucleus. Okay, prokaryotic cells is the other type; they don't. Okay, and they provide those local environments that allow the cell to perform its functions, especially if they're not compatible at the same time. Okay, so it's like the example I used there. We're running your furnace to heat your home, but running the refrigerator to keep your food cold. You can't have those two things going on at the same time. Okay. All right. Everybody okay with that? All right, so compartmentalization of cells has allowed at least eukaryotic cells to become larger, okay, because they do things better. It does make them often more sensitive to changes in their environment, okay? They're not maybe as tolerant to extreme conditions as something that's simpler, but they are going to be more efficient under ideal conditions, okay? So to give you an idea, an animal cell, okay, can be, let's say 10 to 80 micrometers in size, right? Whereas a bacteria, which is a prokaryotic type of cell, might only be 1 micrometer. Right? It's much simpler because it doesn't have the same cellular machinery that the animal cell has. But it still has to do all those same basic functions. It's just got to do them all in the same place. So if you want another analogy, it would be like I'm gonna wash my dirty dishes and my laundry and my, I don't know, I'm trying to think of something else that's dirty, my dog in the same water. At the same time, not one after the other either, because I know some of you are going, well, go there, you just wash the dishes first and then the laundry. Now that probably wouldn't work, because if you didn't add those spaghetti, then all your clothes will be red. See how the analogy works? It's not good, okay? If you're trying to do a bunch of different jobs in the same place, they just don't work very well. Sure, are they all gonna be a little bit cleaner than they started? Probably, but I'm gonna have a red dog, okay? And red clothes, which, yeah, that's not gonna work out well. Dishes will be nice and clean. That's probably important. So that's why prokaryotic cells tend to be small. You can kind of see inside here just a little bit. This stringy stuff that you see right here inside that cell, that's its DNA. There's no nucleus in there. So the DNA just hangs out in the same place as cellular respiration and digestion of food occur. I don't want my DNA kept in the same place I digest my food. My food has DNA in it. That can be digested then. I don't want my own DNA digested. That doesn't make very much sense. It's not very efficient. Okay, so they stay very, very small. All right, and then a virus, okay, you can see there, okay, that virus is like less than half a micrometer in size. Okay, they're not cells, that's why they're so small. All right, questions on the importance of compartmentalization? Okay, that sometimes comes up on a unit exam where you kind of have to explain it, and I've given you a lot of analogies, so you might even come up with your own. Okay, that'd be a good way to explain that, is come up with your own analogy. Okay. All right, so parts of the cell and what they do. The nucleus. Let's start with the big thing in the middle. All right. So inside the nucleus is where your DNA is stored. All right. And DNA is used to make everything. If I took any one of my cells and took the DNA out of it, I could put it in a cloning machine and make an exact copy of myself. Everything that would be necessary for me is coded in that DNA. Right? I don't have a magic cloning machine, but it seems like it would be something that would be useful, as it would save me time. I could just put my clone to work. There might be some ethical problem with that. Would he then be a slave? What if I paid him? Would it be okay then? If you kill your clone, is that murder or suicide? Whoa, that's deep. That's a deep one. I have to think about that one. That's morbid, but deep. Okay. Luckily, I don't have a cloning machine. I don't have to deal with any of these philosophical problems. Okay, so we got DNA inside the nucleus. It's all kept in there. It's nice and safe, but it's not organized. Okay, like you've probably seen pictures of like chromosomes, and it looks like your DNA is all kind of wound up in these like X shapes and things like that. It only looks like that when the cell is preparing to divide. When the cell is undergoing normal conditions, the DNA is completely unraveled inside the nucleus. Okay, so imagine taking a ball of yarn and completely unraveling the whole thing and throwing it in a pile. That's what your DNA looks like all the time in your nucleus. Okay? So it's not very organized, kind of like a hoarder's den, okay, in there. But the nucleolus, that part inside, knows where everything is. It can still find it all. Okay? All right. So the nucleolus, okay, is where this protein synthesis happens. So, sorry, the way this is worded, okay, isn't quite right. So from the nucleolus, that's where protein synthesis is controlled from. Okay, so the nucleus stores the DNA, the nucleolus controls the protein synthesis. And we're going to talk about how that works here in just a minute. So the nuclei are always surrounded by a lot of rough ER. Why is that a smart arrangement, do you suppose? What is the rough rough?
Maybe so it can like keep away all the bad things trying to get to it.
It would certainly be an obstacle, yeah. I would say that would be a secondary benefit of having that all there for sure. Okay? What are the little bumps on the rough ER? What are they? Ribosomes. What do they do? They make protein. Okay, so if protein synthesis is controlled by the nucleolus, and most of the things making the protein are really close, does that make good sense? That makes great sense. Okay, that's how natural selection and evolution work. Okay, generally you get things that work pretty well. Okay, put them close together, and it takes less time for things to happen. Okay. All right, so the nucleolus takes your DNA and it makes copies of certain sections of it. Okay, we talked a little bit about this when we talked about how a virus works. Okay, so if your cell suddenly needs to make some insulin, okay, it'll send a message to the nucleus, and the nucleolus will get it, and it'll say, request blueprints for insulin. So the nucleolus starts going through your DNA, and oh, there it is, there's the part that makes, there's the instructions for making insulin. Okay, now, as a smart librarian, the nucleolus doesn't want to lend out its only copy of those instructions, because it only has one copy of those instructions. Okay, so it doesn't go, oh, I trust you with this. That's not a good idea. Okay, so what it does instead is it makes a copy of only one side of the strand. Okay, because your DNA has two strands. So it only makes a copy of one side. It only has to make a copy of one side. And it sends that out. That single-stranded copy is called RNA. Okay, the double strand is DNA, right? Deoxyribonucleic acid. This is just ribonucleic acid. Okay. So, why does it only need to make a copy of one side? You guys remember from Science 9? The sides are complementary. Okay? A always goes with T and C always goes with G. I think that's how it goes. Okay, so they always pair up. So it can make a copy of either side and it'll still be the instructions for making insulin. And that uses less material. I copy both sides if I don't have to. Okay. So it makes a copy of that. So that's what's going on right here. Your nucleolus is making a copy of one side of your DNA, and then it sends that copy out through the nuclear pores, those special little gates that open and close. Okay? That RNA will then join up with a ribosome. The ribosome can read it, and it'll put together all the raw materials that are listed in the order they're listed, and that'll make a protein. Okay? You will not have to describe that process in detail to me ever. Okay? Just explaining how the process works so you know what the nucleolus does. Okay? You can tell me the nucleolus copies DNA or controls protein synthesis, and that's good for its function. That's how it does it. Okay? Copies your DNA, sends the copy out so the copy can be used to make protein. Okay? That's how your cells make protein. And it's the ribosomes that actually put it together. All right. Questions on that? All right, so we've now talked about what the nucleolus does, and we've talked about what the nucleus does, and we've talked about what the ribosome does. Okay, ribosomes make protein. Okay. All right. Endoplasmic reticulum. Okay, there are two kinds of endoplasmic reticulum, rough and smooth. The rough ER is actually quite uniform, like it's layered. It looks organized. It's covered in the ribosomes. The smooth ER looks quite random, just like squiggles everywhere. In fact, you probably won't even see it through the microscope when we look at the cells. You will see the rough ER though. Okay? The rough ER is actually visible in a cell. But because the smooth ER doesn't have any ribosomes on it, it ends up usually just being transparent and we don't notice it. Okay? So you've got your nucleus and then you've got your rough ER around the nucleus. Okay? And then the smooth ER is usually kind of next to that. Right? All these little black dots here, okay, each one of those is a ribosome. You're not going to see the ribosomes under the microscope. Okay? That picture right there, okay, that is from an electron microscope. Okay? That is magnified probably several thousand times. Okay? Our microscope at best is 400. Okay, so while you might see the rough ER because it's covered in ribosomes and that makes it easier to see, you're not going to be able to see individual ribosomes. They are far too small. Their existence wasn't even known until the electron microscope was invented. Okay, so they're really, really small. Okay, so if we're looking at what the ER does, okay, start with this well, we'll actually start with the rough ER. It's not listed here, but you'll want to write this. The rough ER transports protein. Now, the smooth ER has a lot of jobs. Okay? Even though it's something we're not going to see through the microscope because it's pretty much transparent, it actually fills up a lot of the volume of the cell. Okay? So within the membranes of the smooth ER, lipids are synthesized. What are lipids? What's a more common name for lipids? Fats. Yeah. Okay? So fats are synthesized. Now, as soon as you say fats, people are like, fats are bad. Okay? Actually, fats are good. They not only taste good, but they're good for you. Okay? Not in the amounts we consume them in the North American diet, but they are what make up all membranes. Every single membrane in your body, cell membrane, membrane of an organelle, the nuclear membrane, all of them, they're made of fats. Okay? So we gotta have fat in our diet because that's the raw materials for making cells. Okay? So fats are really important. Okay? We have to make certain fats to make membranes, and that's one of the jobs, probably one of the more important jobs of the smooth ER. Okay? That stuff is used to repair membranes. Membranes get damaged all the time because their job is to separate in from out. Okay? So stuff runs into them, chemical things can happen and damage parts of it, they have to be repaired. Okay? So lipids are synthesized for building and repairing membranes. So that's one job of the smooth ER. Okay? Another job of the smooth ER is to break down complex carbohydrates. Okay? Complex carbohydrates are also known as starches. What kind of foods would you get starches from? Potatoes are very starchy. Pasta, rice, okay? Basically your big carbs, okay? They're made of complex carbohydrates. Your lysosomes don't break that down. Your lysosomes break down things like fats and proteins. Complex carbohydrates get turned into glucose easily by the smooth ER. Okay? All you have to do with a complex carbohydrate is cut it up into pieces and it's magically glucose. Okay? So if I have fructose, for example, which is a very common sugar, sucrose, another very common sugar, they're basically like 2 or 3 molecules of glucose stuck together. Okay? So they're very easy to break down into glucose. So that happens. Okay? Complex carbohydrates are broken down. Okay? And drugs and poisons get detoxified in the smooth ER. Okay? You want to have drugs and poisons and things that are bad in a place where they can't make contact with other parts of the cell. So if you keep them in the smooth ER, that makes good sense for a couple of reasons. It's capable of breaking most of them down, and its job is to repair membranes. So any damage caused by them, it can repair itself. Okay, so it makes good sense to have that go on inside that organelle. Okay, so we can detoxify drugs and poisons. Not all things, obviously, okay, or maybe not all things quickly. Okay, some, poisons are easily detoxified, and others are not. Okay, so complex stuff would take longer. So if I was looking at a liver cell, would liver cells have a lot of smooth ER in them? Yeah, because your liver's job is to clean your blood. Okay, and one of the things it cleans out of your blood is toxins. Okay, it's what can cause a hangover. Okay, if you have a hangover, you often feel sick to your stomach because what's happening is your liver is breaking down the alcohol, and the byproducts of that breakdown are nauseating. okay? They irritate your small intestine, and that's where they go. In order for your liver to get rid of a lot of them, it sends them down the bile duct and into your small intestine, which then gets irritated, and you feel icky, okay? And you'll often have a headache, which is often caused by an imbalance of toxin and things like that in the blood, right? That's typical symptoms of any type of poisoning, okay? And a hangover means you've had a little bit of alcohol poisoning, okay? Any type of poisoning is going to give you similar symptoms to that, okay? And that's the result of your body doing its job, trying to get rid of these awful things that you put in it, okay? And trying to make you feel better. All right, and the last thing that can be done in the smooth ER is hormones can get produced there, okay? One kind of hormone in particular, fat soluble. Okay, because there are different kinds of hormones. So fat soluble hormones can be produced there. Smooth ER pretty important. Does a lot of jobs. How many of those would you have to list if I asked you what the function of the smooth ER is? One or two. Okay, there's 1, 4 there, 5 there. Okay, 1 or 2 is good. Okay, as long as you remember a couple of them, you're good. Okay, and as I was saying before, the reason we want to perform those kind of tasks there, like making hormones, cells usually make hormones not for themselves, but to be exported. So if you make them in the smooth ER, smooth ER has access to the membrane to get rid of them. Same with the toxins, right? You can detoxify them and get rid of that stuff and get it out quickly. That's why we do those jobs within the smooth ER. Okay? Also, it makes lipids, which are needed at the membrane. So being attached to the membrane, also good for that. Sends them out there quickly. All right, questions on the smooth ER and rough ER? Okay. Okay, and we already talked about that. Lipids get moved quickly. So if we're looking at kind of a different diagram of it, so this is an electron microscope picture of that same thing. So this is the rough ER here on the right. Okay, you can see all the ribosomes. You can see it's very organized and layered. This is what the smooth ER looks like here on the left. It's just random. Right? It's literally like just a snarl, okay, of stuff. Okay, the Golgi apparatus. Like we said yesterday, we sometimes treat it like it's the garbage man of the cell. Okay, it is responsible for packaging materials for export. Okay, to take them to the cell membrane. Okay, anything that's going to get out of the cell, okay, we call that exocytosis. We'll talk about that later. But the Golgi is kind of responsible for getting things to the point where they can undergo exocytosis. Okay, so a lot of the products from the ER go to the Golgi and then they get taken out of the cell. Okay, so that could be things like the hormones that were produced, okay, proteins that were produced, okay, stuff like that. And then of course any waste products as well. So the Golgi has this weird shape, okay, but it has a reason for that weird shape. It has 2 faces or 2 sides. It has the cis face and the trans face. You don't need to call them that or even remember that. Okay? It's basically receiving and shipping. Okay, so the stuff comes in on one side, the Golgi will process and package it and ship it out the other side. Okay, so when stuff comes into the Golgi, it's often exposed, okay, or open, right? So this is our cis face right here, okay, on this side. So you can see that there aren't any bubbles on this side. Everything is coming in open. Okay? As it goes through the Golgi apparatus on the trans face, you can see there's bubbles everywhere. Okay? Inside of those bubbles, we call them vesicles, okay, is all the stuff that came in on this side. So as it goes through the Golgi, it puts it in a bag, okay, and sends it out, or puts it in a box and sends it out. Right? It's in charge of shipping and receiving. Okay? It brings the stuff in, it packages it, and it makes sure it's going to the right place. Okay? That sort of makes sense? Right? So it's like the Amazon sorting facility, okay? Kind of like that. All right, and we can see that kind of happening here. Okay, so we got stuff coming in on the cis face, okay, and it comes out of the trans face in a vesicle, and then that vesicle will merge with the cell membrane and just pop whatever's in it out into the fluid, okay, blood, whatever it happens to be. All right, so the Golgi transports material out of the cell. Okay, that would be kind of its job. If you were going to list something on a quiz, I'd say that would be a good description of what the Golgi does. All right, so lysosomes, we talked about those yesterday, they digest food. They're only going to be in what kind of cell? Animal cells. Okay, so that's an important thing to remember. Everything else we've talked about is in both. Okay, but the lysosome is only going to be in animal cells. All right, and its job is to digest material. Okay, so lysosome kind of looks like this. Okay, it's got hydrolytic, that's digestive enzymes. Okay, and its membrane there, and then materials can be digested inside of there. Right now, there's two kinds of lysosomes, we'll say. Right, there's this is actually not labeled correctly. There's peroxisomes, that would be this part. Okay? A peroxisome only has digestive enzymes in it. Okay? When it merges with a food vacuole that has food in it, then it becomes a lysosome because digestion is happening. And so it's only a lysosome if it's actually digesting something. If it just has the enzymes, it's a peroxisome. It's an empty stomach. So the lysosomes digest food, but that's not all they do. Okay? They also help with recycling. Your cell is the ultimate recycler. Right? If an organelle breaks down and it's just not working anymore, the cell will eat it. Okay? It's called autophagy. It means kind of eating itself. So it'll digest that, break it down into its basic materials, and then those basic materials can be used to make a new one. Okay, or to do other things. Okay, it can always produce another organelle, okay, on its own. Okay, but that's kind of what one of the other jobs it does. It's kind of recycle things. Its last job is one it hopes it never has to do. Cells have an ability to detect if they're malfunctioning to an extent. Okay, it's not perfect, but there's a mechanism in place. Okay, and if a cell detects that it's malfunctioning and could possibly be malignant. You guys heard that term before? Okay. What do we usually use the term malignant in reference to? What disease? Cancer. Yeah. Okay. So if a cell becomes malignant, that means it could become cancerous. The cell will, if it detects it, will destroy itself. And the way it does that is it ruptures all of the lysosomes inside. And the cell will literally eat itself to death. Okay? All the digestive enzymes will get out and they'll digest the cell from the inside out. Okay? But then the cell won't become cancerous. Okay? A cell becomes cancerous if it becomes damaged and starts dividing out of control. Okay? And that's the problem with cancer, is that it's your own cells. Okay? So what happens with cancer is a cell gets damaged and starts to divide. It divides way faster than it's supposed to. And you end up getting a lump of cells in one spot. That's the tumor. Okay? But your body doesn't always recognize there's anything wrong. It's like, oh, well, that's us. Those are our cells. They belong here. Except they don't. With cancer, what happens is the cells get growing and growing. That tumor gets bigger and bigger and bigger, and it starts requiring a lot of raw materials to keep that process going. So whatever organ it's attached to is no longer getting the nutrients and the blood supply and all of that kind of stuff that it's supposed to, and the organ it's attached to slowly dies because the tumor is what gets fed. Okay? So it's really insidious. It's hard for your body to fight that because it doesn't recognize that there's anything to fight. It's not like when you get sick, you have a bacterial infection, your body knows that stuff doesn't belong here, kill it. Okay, a virus, ooh, you gotta get rid of that. We recognize that's foreign. It doesn't recognize a cancer cell as foreign. Okay, and so that's why we have to do other things to get those cells under control. So we have things like chemotherapy. Okay, chemotherapy it's poison. Okay? It's why a person on chemo feels awful, why their hair falls out, why they become immune suppressed. Chemotherapy kills all rapidly dividing cells, which is why it's good to kill cancer with. But other rapidly dividing cells are hair follicles, stomach lining, and immune system. And so those often get depressed. People that that are having chemo will often get ulcers because their stomach lining doesn't replenish itself fast enough. They lose their hair, okay? And they can get sick very easily. They have to be protected or isolated oftentimes because they get immune suppressed. Does that sort of make sense? Okay, so one of the jobs of the lysosomes, long story short, is that it can destroy the cell. It's a self-destruct mechanism, okay, in an extreme case. All right, questions on the lysosome? All right, the mitochondria. These are in both plant and animal. This is where cellular respiration occurs. So where glucose gets broken down into carbon dioxide, water, and energy that the cell can use. Okay, so the mitochondria actually has two membranes. Okay? And the reason for that is there's space between the two membranes, and that space acts like an insulator. Kind of like why your house has an outside part to the wall and an inside part to the wall, and what's in between the inside and outside? Insulation. Okay, so the mitochondria is kind of built that way because the mitochondria gets hot. That's where glucose is being broken down and heat gets produced in that reaction. Right? So it has this double layer to kind of insulate the rest of the cell against itself, because the rest of the cell doesn't want to be hot. Okay? Just like in the same way, if you get a fever, how do you feel? Like bag, right? Like if you got a fever, you're tired, okay? You feel like you're sore, okay? All that stuff, because your body is set to work at 37. Okay? If you run a fever, you get your body to 39, 40. The stuff that's supposed to happen in your body doesn't happen. Okay? It doesn't work at higher temperatures. Right? Well, the same thing is true in an individual cell. We don't want these things to get out of control and get the rest of the cell hot, or the processes in the cell won't work right. Okay? This is the importance of compartmentalization. We keep that compartmentalized. We don't let it get anywhere else, okay, to protect the rest of the cell. Now, if you're running a fever, it's because of these. Okay? Your body runs a fever to try and slow down the process of whatever is making you sick. Okay? So if you get a bacterial infection, one of the first things your body will do is run a fever, because the bacteria also don't work well at higher temperatures. And it's a natural response of the body to go, okay, well, let's elevate the temperature and see if we can slow that stuff down and let the immune system take care of it. Okay? But if it gets out of control, fever can be bad for you too. Okay? A low fever, that's okay. It helps your body out. Okay? It doesn't necessarily feel good, but it helps the body out. All right. So basically what we need to know about the mitochondria is that is where cellular respiration occurs. Cellular respiration happens. Okay, on a quiz, would cellular respiration be good for the function? Even on an exam, it would be good for the function. That's exactly what it does. Okay, chloroplasts. Only going to be in plant cells, okay? One word for the function, what is it? Photosynthesis. That's what the chloroplast does, okay? Inside the chloroplast is chlorophyll. We talked about those yesterday, okay? The light hits it, gets the electrons in the chlorophyll excited, they get away from the nucleus, they fall back down later, and the energy gets released and makes sugar, okay? All right, so that's all we need to know about the chloroplast, okay? Chloroplast carries out photosynthesis, pretty simple. All right, some other organelles. I found a good picture of the cell membrane. Remember yesterday I was like, you can never find a picture that shows what the cell membrane is like. It's like Jell-O salad. This is what the cell membrane looks like, okay, right up here. So all these little white circles with the little tails on them, those are fats, okay, those are lipids. So the majority of the cell membrane is made of fats and lipids, okay. In the matrix of those lipids, okay, are other things, okay, like proteins, oligosaccharides, okay, cholesterol. Cholesterol is actually an important part of the cell membrane. Again, we don't want as much of it as we typically consume, okay, but it is an important steroid that is part of your cell membranes. Okay, so you got globular proteins, there's sugars, phospholipids, all those kind of things. There's lots of things, okay, in that membrane that make it work. Some of these are even receptor molecules. They're like antennas, okay, on the cell membrane, and they receive the chemical signals your body can send, hormones. Okay? And there's actually one antenna for each signal. Okay? They don't have antennas that accept multiple signals. There has to be an antenna for each signal. Okay? So there'd be one on each cell that would be for adrenaline, one that would be for insulin, one that would be for estrogen, one that would be for testosterone. Okay? Any chemical signal that your cell can receive, there's going to be something embedded in the membrane to receive it. Okay, so cell membrane. If I was going to write 2 words for the function of the cell membrane, those words would be these: endocytosis and exocytosis. That is movement in and out of the cell. Endocytosis is in, exocytosis is out. Right? So the thing I don't want to see is protects the cell. Don't write that. Okay? I don't want to see that as the function of the cell membrane. Cell membrane is not a border guard. Okay? It is not like checking people's ID and making sure that they're okay to come in. Okay? It's not like that. It's selectively permeable. Some things can get through and some things can't, but it makes mistakes. Okay? It doesn't do a great job of protecting the cell. It just kind of controls what gets in and out. Okay, lets things in and out, but it's not always perfect. Okay. All right, the other thing I told you, I can never find a good picture. I found a good picture. The cell wall. Remember we said it always looks solid on the diagram, but I told you it's actually more like a frame. It's like a house that's being built. You could still walk through the walls even though they can hold up the house. This green part here, that's the cell wall. Okay, so it's quite clearly not solid. Okay, it's a frame. It's got holes in it, right? So it also doesn't protect the cell. All it does is support the cell's shape, okay? Because it's full of holes. Anything can get in or out of it, just like you can. It doesn't do any good to put a door on a house that's in the middle of being framed. Okay, I don't have to unlock the door. I can just walk through the walls. Okay, that's what the cell wall is like. All right, so the cell wall, okay, is the cellulose fiber frame of the cell that keeps the shape and supports the cell. Okay. You don't have to remember it's made of cellulose, although we did have a little chuckle about cellulose is that plant fiber we can't digest, okay, which is why, corn doesn't get fully digested when it goes through. Okay. It's got a lot of cellulose in the husk. All right, the water vacuole. Which kind of cell is that in? Just in plants. Okay, so the plant, the water vacuole uses turgor pressure, that's the pressure of the water inside, to support the cell. Okay, it pushes against the cell wall, so those two work in concert with each other. Yesterday I showed you what that plant looked like because I hadn't watered it. Okay, I watered it yesterday. Okay. These ones might have been too far gone. Okay? The rest of the leaves are starting to perk up already. Okay? Those water vacuoles are filling back up. Okay? Looks like there's a few here that maybe were too far gone. Definitely too far gone. Okay? But the rest of them are starting to fill those vacuoles again. They're getting rigid and holding up. Okay. Centrioles. Those are only in what kind of cell? Animal cells. All right, so centrioles work like this. Okay, when the cell is getting ready to divide, all of its DNA gets condensed into chromosomes, which can look like this. Okay, and they all line up right along the middle of the cell. Okay, then the centrioles go to either end. So they start out like this and they split and they actually replicate while they're moving to the two poles. Okay, Okay? From those poles, they send out these fibers that attach to the chromosomes. And then just before the cell actually splits in half, the centrioles pull the chromosomes apart, making sure that the right amount of DNA gets into both sides. Okay? Then the cell can split, nucleus reforms around them, okay? And it's good to go from there. Right? So the centrioles, that's way more detail than you'll need to know. Okay? Centrioles basically aid with cell division. Okay? If you put helps cell divide or helps with cell division, that would be good enough. Okay, and lastly, I didn't have a picture of it in here for some reason. Microfibers and microfilaments, okay, those are going to be present mostly in animal cells. We sometimes call that the cytoskeleton. Okay. Cyto means cell, like skeleton of the cell. They just support the shape of the animal cell because obviously it doesn't have a wall or a vacuole that does that job. So for microfibers and microfilaments, okay, that basically like hold cell shape, something like that. Okay. All right, questions on any of those other things? All right, that's everything that's on your cell diagram. Okay, so now you know what everything looks like and what everything does. And again, I'm not ever going to ask for a like detailed paragraph answer for what the role of the smoothie R and cellular function is. Okay? I'm going to say, tell me what it does. And you can say, breaks down complex carbohydrates, detoxifies poisons, makes fat soluble hormones. Okay? That's all the detail that I would be looking for. All right, let's take a little break before we start talking about transport within the cell, because that's the next thing here. All right, so the next thing we got to talk about is cells obviously have to be able to move things from one place to another within the cell. They also have to be able to transport materials across the cell membrane because stuff has to get in and out. Right? You've got your wastes that you want to get out, or anything that you've produced that needs to get sent elsewhere needs to get out, and then obviously all the raw materials for all that stuff has to get in. Right? So there's a number of different processes that the cell uses in order to do that. Okay? What we'll start with is the process that the cell uses to transport material from place to place within the cell. Okay, because it only uses one process to do that, and it's free. Okay, the process is called diffusion. It's simply a natural byproduct of the fact that all molecules and atoms are moving. Okay, so anytime if I'm looking at if I could actually see the molecules of air in this room, they would be moving all over the place. Okay, and that's true of any particle unless it's at absolute zero, okay, where all molecular motion ceases. Okay, but that's not the case. So there's always molecular movement, and that movement is always going to cause molecules that are closely or highly concentrated to move away from each other. Okay, because while they're trying to move, they're going to run into each other, and that's going to cause them to bounce and move away. Okay, so the process of diffusion is the natural movement of material from an area where it's highly concentrated to an area where it's lower in concentration. Okay? I'm going to explain this to you with an analogy and you're never going to forget it. Okay? Here's the analogy. Diffusion is the process by which a fart is detected by others. Okay? So, if, let's say, I was to fart right here in front of the classroom, okay? Those closest to me would be most affected by it because close to me, the fart molecules would be very concentrated, okay? But as they move away from me, they become less and less concentrated. So, if you're Kate over there in the corner, you might not even detect the fart because by the time it gets there, it has diffused out and gone into such a huge area that the concentration is so low as to be virtually undetectable. Okay, but poor Cassidy is right next to me, and it would be highly unpleasant for her. Okay, that sort of makes sense. Okay, that's the process of diffusion. All things do that, especially smells. Okay, that's the thing that we probably have the most experience with is knowing that, yeah, if I'm next to something that smells really bad, my natural reaction is to move away from it because I know the smell won't be as bad the further away I get. And that's because as it diffuses away, the space between the molecules gets bigger, the concentration goes down. Okay? So the same thing could happen if I put like dye into water. So if you put a drop of food coloring in a glass of water, right where the food coloring goes in, it's very dark. Okay? But in a very short amount of time, it spreads out over the entire volume of water in the glass and isn't nearly as dark by then. Okay? That's the natural process of diffusion. So cells use that to transport materials. It's really good at doing that, but it's got limitations. Okay? Diffusion is like free shipping. Okay? I mean, if you've got Amazon, let's say, and you've got Prime, you can get stuff in 1 or 2 days, but you pay for that. Okay? It's not free shipping, you pay for it, so you get it faster. Okay? Free shipping, you gotta wait a little bit longer. Okay? But you'll still eventually get it. That's kind of diffusion. Diffusion is free, but you get what you pay for. It's gonna take longer, especially if it has to go really far, and it's not targeted. So if I've got a part of the cell where something is being produced, it's going to be in very high concentration there. It's just going to randomly move away from that to areas of lower concentration. Now, that generally works because usually where it's in low concentration is also where it's needed. So it's generally going to move in the right direction, but there's no guarantee. Okay? So it generally works, especially in an area as small as a cell. Okay? If we're talking about the food coloring in the glass, that's not really, it takes quite a while for that to all spread out and be all through the glass because diffusion is slow over large distances. And we're talking large on the scale of molecules. Okay? A glass of water is a large distance on the scale of a molecule, but a cell is not. Okay? What that means though is there's an upper limit to the size a cell can reach. If you get too big, your free shipping doesn't get stuff places fast enough, okay? Or accurately enough, and then the cell can end up like having issues with starvation and things like that. Okay, so diffusion is passive. Okay, so any type of transport that is passive, okay, means that the cell doesn't use any energy. It's free. It's literally free for the cell. It doesn't require the cell to do anything except let it happen. Okay, that's good. The cell doesn't want to have to pay for things, right? The other downside of that is it's going to happen. The cell also can't stop it. Okay, it's just a process that's going to happen. Passive processes are not controlled. They just happen. All right, so the simple rule for diffusion is, okay, in the absence of another force, like a current or something like that, which wouldn't be present in the cell, a substance will diffuse from where it is most concentrated to where it is less concentrated or down the concentration gradient. So that second point basically explains how diffusion works, okay, and diffusion is passive. Okay, which means it requires no energy, which is why a cell uses it to move material from one place to another. Okay? It works really well over the distances associated with a normal sized cell. Okay, questions on diffusion? All right, so what we're going to talk about here are kind of the big three methods of cell transport. Diffusion is number one, okay, because it works everywhere inside the cell. In the fluids outside the cell, in your own body, okay, everywhere, in the air, in the water, all over. Okay. The second method of cell transport is called osmosis. You guys have probably heard of this, okay? Especially if you have reverse osmosis water, or you like drinking reverse osmosis water. Okay? That process is exactly as it sounds. It's the actual opposite of real osmosis. Okay? Osmosis is movement across the membrane. So now we're not talking about movement in the cell anymore. We're talking about movement across the cell membrane. And the only thing that osmosis moves is water. Okay? So that's kind of its defining characteristic. If I was describing a situation on a test, okay, and asking you, identify and describe the cellular transport process being described here, okay, and it said it's the movement of water, automatic, okay, that's the only thing that osmosis moves is water, okay, so osmosis is the passive transport of water, okay, and it is across the membrane. Okay, and it does this to balance the concentration of salts on either side of the cell. Okay, so what happens if you put salt on a slug?
It dies.
Yeah, it does. But as it's dying, it shrivels up. Okay, and the reason that happens is that's osmosis. Okay, if you put salt on the outside of a slug, because it doesn't have skin like we do, it can't control or prevent osmosis, which is a passive process, okay, from happening. And so all the water inside the slug rushes out through the slug's skin towards the salt to try to make the concentration of salt outside and inside equal. But there's so much salt on the outside of the slug that that's impossible, and the slug just shrivels up and dies. Okay, because it can't stop this process. Nor can we, okay? So, if you were in a lifeboat in the middle of the ocean, should you drink the water? No, the ocean is the world's toilet. Don't, oh, besides that, it's salty, okay? And salt water, if you consume it, you guys seen Finding Nemo, right? All pipes lead to the ocean. It's the world's toilet, don't drink it, okay? But also, it's salty, okay? And if you consume all of that salt, suddenly your blood gets highly salty, and it starts pulling water out of your tissue, out of your muscles, okay, out of your, your skin, all of the internal parts of you it starts pulling the water out of there because it's trying to balance the concentration of salt in your blood with the concentration of salt in your cells, which causes you to shrivel up, like almost literally, okay, but you can become dehydrated very quickly. Okay? There's so much salt in ocean water, okay, that process will occur very, very fast. In fact, for every liter of salt water you drink, you'll lose 2. Your body will have to flush another liter in addition to that out through your kidneys, okay, in order to get the salt out. Right? You're actually better off to drink your own urine for the first couple of days. There's actually less salt in that than in ocean water. Survival tip for you. Hope you don't ever have to use that. Okay, but yeah, osmosis is this passive movement of water in response to salt. Okay, similarly, if I took that slug and I put it in distilled water, what would happen to it then? There's no salts at all in distilled water, but there's salts inside the slug. Which way is the water going to move? In to the slug. The slug will swell up. Okay, put salt on the outside of the slug, the water goes to the salt. Put the slug in something that has no salt in it, the water goes into the slug because that's where the salt is. Okay, this is a passive process. We can't stop it. It just happens. Okay? It's also why you can put salt on meat and dry it out and cure it and make jerky. Okay, you put salt on the meat, the water moves out of the meat towards the salt, dehydrates the meat. Okay, and then it doesn't rot nearly as fast. Okay, plus the salt also prevents things from growing there. Okay. All right, so osmosis, passive movement of water across the membrane, okay, to gain equilibrium, okay, or you might want to say balance salt concentrations. Okay, and I don't mean like just table salt, I mean any ionic compounds, any salts, okay, that could be there. Okay, this is a lab you'll actually do, I think in Bio 30, when you're doing the kidney. Okay, you'll use a special type of tube, okay, that's just bent into a horseshoe, and you put a piece of sausage casing across the middle. Okay, sausage casing is made of intestine. Okay, so it's basically just skin. Okay, and so you put salt water on one side, and you put like just regular tap water on the other, and over the course of time, the water will go across the sausage casing towards the saltier water. Okay? And so what you'll end up with is less water on this side, more water on this side, but an equal concentration of salt on either side. Okay? So it'll keep moving until the salt concentration is balanced, okay, on either side, which means one side will go down, the other side will go up. Okay? Making some sense? Okay? This is why, like, if you're really dehydrated, right, and you go into the hospital, they put you on an IV, okay? Is it just water? It's got sodium chloride, just regular table salt in it, because that helps speed the process of osmosis to happen. Okay? It puts a little bit of extra salts and ions into your tissues, okay, allowing that osmotic balance to get set up faster. Okay, so it's never just distilled water in an IV. Okay? It's always got usually sodium chloride in it, okay, to try and get that going. It's why things like Gatorade, Powerade, any of those sports drinks, they actually have ions in them to help reset your osmotic balance and help you hydrate faster. That's kind of the science behind it. Okay. So if we had these cells here, so this is kind of the same thing as the slug I was talking about. If I put a cell, okay, into what we call isotonic, iso means equal, okay, so I put it in a solution that's got the same concentrations of salts as the cell, equal amounts of water move in and out. Okay, everything's balanced, everything's happy. Okay, if I put it in hypertonic conditions, that means the solution outside the cell has got more salt, then water moves out of the cell and towards the salt. It's not the salt that moves, it's the water. Okay, so the water moves out of the cell into the salt and the cell shrivels up. Okay, if I was to put it in distilled water, where there are no salts, okay, then there'd be more salt in the cell and the water rushes into the cell and the cell will burst. Okay, because it'll never get those concentrations equal. There's no salt outside, so it would be impossible to ever get them equal. It'll just rush in until it bursts. Okay. All right, questions on osmosis? All right, the last one is the only one that's not passive. And thus its name is active transport, because it's the opposite of passive. Okay? It's active transport. Your cell actually expends energy to use this process. So if your cell is going to use energy to use this process, what it needs is pretty important. Okay? It's not going to do this if it's not trying to get something in a hurry. Right? So active transport is the pumping of solutes against their gradient. Okay? This pumps water uphill. Okay? It makes things go from where they're in low concentration to high concentration, the opposite of what diffusion does, the opposite of what osmosis does. Okay? So it's going to make things go the other way, and it's going to actively pump them that way. Okay? Does that sort of make sense? So this is one that's used by your muscles and one that's used by your nerves. Okay? In order for your muscles to activate and relax, okay? Activate and relax, sodium and potassium have to be actively transported across the cell membranes alternately. Okay? So in order to get your muscles to contract, it goes one way, and then to get them to relax, it goes the other way. Okay? So that's active transport. That's why you can get a muscle cramp if you get dehydrated. Okay? If you get dehydrated, your muscle won't relax. Okay? It just gets into a spasm and into a cramp, and you can't relax it. That's because you don't have enough water there, okay, for that osmotic balance to get reset, and your active transport can't work. Okay? Does that sort of make sense? All right, so active transport pumps solutes against the gradient. Okay, it uses energy. The cell has to expend energy to do it. Okay, meaning it actively pumps a substance across the membrane. Okay, now the key part to making this work is special structures embedded in the membrane. Okay, we said that the membrane is like Jell-O salad. Well, some of those things that are in it are called carrier proteins or transport proteins. And when energy is applied to them, they change shape. Okay, so if the cell is going to expend energy to do this active transport, what that energy does is changes the shape of the carrier proteins. So in normal conditions, the carrier protein looks like this. It's open to the outside, okay? And ions can move in to the gap. When the cell expends energy, the cell's protein shape changes and closes the gate, now pushing that material into the cell, where it's actually higher in concentration. So the cell has to do this because naturally the stuff wants to go the other way. It wants to go from high to low passively, okay? But the cell's like, nope, we need lots of that, okay? We're gonna pile that in here. We're gonna keep pumping it in, okay? And that's gonna use energy. So the thing you need to have, in addition to what's here, is the mention of carrier proteins, energy, and the fact that that changes the shape of the proteins. That's what causes the pumping, okay? Okay, so those are the big three methods of cellular transport. They transport the bulk of materials for a cell. We're going to talk about a couple of others tomorrow that involve changing the shape of the membrane, basically engulfing things, and just like swallowing them up and bringing them in. That's different, okay? So we'll talk about those. They're never going to be tested in like a written response item. They might be a multiple choice, whereas these would definitely be more important, so they would be more written response kind of questions. Okay? All right. I'm going to leave it there. That's a natural breaking point before we get to phagocytosis, endocytosis, exocytosis, all that good stuff. Okay? Tomorrow, you have a quiz. It'll post at 3:20 today. It'll be on, I'm not going to do the labeling of the cell yet, because I just taught you the functions today. I'll give you till next week to learn that. Okay? So this will be on modes of nutrition, cell theory, okay, stuff like that. That's what tomorrow's quiz will be on. I haven't decided. Probably not. Because I don't see it.